Hello students, welcome to EPG Pathshala. I am Natika Walia Chabra from Cherry. Today we are going to talk on module secondary metabolites under the paper Plant Biotechnology and Crop Improvement. So let us understand what we are going to cover in this module. We are going to talk about what are secondary metabolites, whole plant system versus in vitro system, the process, how do we carry about secondary metabolite production. Then we are also going to talk about the factors affecting secondary metabolite production, and finally bioreactors and hairy root culture. So let us first understand what are secondary metabolites. But first of all, I'm sure you must be knowing what are primary metabolites. Primary metabolites, they are the biomolecules which have a physiological role in the processes of a plant, right? And they are involved in both growth and development of the plant system. And the secondary metabolites, they are derived from primary metabolites, and to define them, they are the biomolecules that are involved in that are not involved in growth and development processes of the plant and are mostly produced under conditions of stress. And the stress can be biotic as well as abiotic. Now they have their role has been mainly of protection in the plant, but a lot of research has gone into it and we have found lot many uses, and they have varied application in large number of fields such as pharmaceuticals, feedstock, agriculture. They're also used in flavors and fragrances. They have antimicrobial activity also, and in some plants, the secondary metabolites are used as food additives. Now, just to brush up your knowledge, the secondary metabolites can be broadly categorized into three. These are phenols, terpenes, and nitrogen-containing compounds. And under each of these categories, there are many biomolecules which are categorized under them. For instance, in phenols, we have phenolic acid, coumarins, lignans, stilbenes. Then we have tannins and lignans. Under terpenes, we have plant volatiles, we have cardiac glycosides, then we have carotenoids and sterols. In nitrogen-containing compounds, now we are going to talk about what are the sites of production of secondary metabolite. Secondary metabolites can be produced in the entire plant body, or it can be very localized. Like it can be produced either in the flowers or leaves, bark or roots. Now, how do we carry out commercial production of secondary metabolites? There are two ways by which we can do it. First, we can either produce it in the whole plant. We can use whole plant as our factory, or we can do it under in vitro culture. Now, if we are using whole plant system, we would either have to grow the plants in the fields, or we have to harvest them from the wild. Now, this is a problem, because if we are growing the plants in the field, we would be using our land which is otherwise used for producing food for production of secondary metabolites. And as we know that we have to feed a large population, it would not be a viable option. And if we are harvesting the plants from the wild, the perpetual harvesting of plants from the wild would lead to their extinction. In addition, if the plants are growing in the field, or either they are growing or either we are harvesting them from the wild, they would be exposed to the vagaries of the nature, and as a result, the yields would vary. So in order to have consistent yields, it would not be a good option. Exploring the second option, that is in vitro system, it is like it is a continuous reliable source of secondary metabolite production, because there is no season specificity. We can scale it up as much as we want, and there would not be any danger of crop failure. And in addition to this, under in vitro condition, there is production of some novel compounds which are otherwise not found in nature. Now, this in vitro system also has certain drawbacks. Like that most of the natural products, they have been substituted by the chemical analogs or synthetics because the cost of production is very high. That the high value specialty compounds, they are actually produced in very less quantities. So to have a commercial yield, a lot of research needs to be done and the production needs to be increased under in vitro condition. And this in turn raises the production cost. So, and besides that, it has also been very commonly observed in cell cultures that the cells which are under culture, they undergo mutation. So they are not stable. So because these are very genetically unstable, we constantly needs to have a pipeline of cells which are homogeneous, which are homogeneously producing secondary metabolites. They're all uniform genetically so that the secondary metabolite production is uniform. Now students, we will understand the process. How is a secondary metabolite production carried out? We can break the process into 5 broadly. Like initially what we are going to do, we are going to select an elite genotype which would be high secondary metabolite producer. So we can carry out chemical profiling of the genotypes, and after that we can select the genotype which is a high producer. Thereafter, in the second stage, what we will do, we will initiate cultures using a high concentration of anoxin, which is 2,4-D. We know that 2,4-D helps in cell division. So we are going to culture our X plants, which can be a leaf tissue or any tissue from which it is known that it has high concentration, it produces high concentration of that particular secondary metabolite. So we induce that tissue to produce calli. So once we have initiated the cultures, we keep it under idle conditions and there is appearance of callus. We take that callus and take it on to the next stage. We raise a cell suspension culture and incubate the cell suspension culture for almost, you can say, 3 weeks. Thereafter, we are going to draw an inoculum from that cell suspension culture and put it on a selection plate, which would have all the same composition as the suspension culture, but it will be in a semi-solid form, so that the individual cells can grow and form colonies. The next stage would involve screening of the individual colonies. We are going to test which colony is high producer, because we know in a plant body also there is genetic heterogeneity. So which part of the tissue is actually producing callus, that would determine the secondary metabolite production, whether it is a high producer or it is a low producer. So we are going to select a colony which is a high producer. So once we have selected that, we are going to move to the next stage, which is biomass production. We are going to optimize the media so that the biomass increases over a period of time. Now, once we have reached the optimal stage, we have to undertake secondary metabolite production. As you will come to know in the subsequent part of the lecture, that the two processes, the biomass and the secondary metabolite, they are inversely related. So in the second phase, which is the secondary metabolite production phase, we have to do media modification and we have to change the culture condition in a way that it stops further growth and starts producing secondary metabolites. So once we have obtained sufficient quantity of secondary metabolites, the next stage would be extraction, because the secondary metabolites, they are produced mostly intracellularly. So we have to adopt mechanism so that it is either released, so it is released in the medium and from there it is extracted. Factors affecting secondary metabolite production. The production of secondary metabolites under in vitro conditions is greatly affected by the culture conditions. It has been observed that the growth, when the cells are growing actively, at that point of time, the secondary metabolite production is either very less or it is not there. So we can say that growth and production of secondary metabolites, they are inversely related. And as we can see in this slide, the secondary metabolite production, it takes place in the stationary phase, and during this phase, what happens is the media is depleted, and as a result, the cells stop growing further, and the cytodifferentiation, and there is induction of enzymes which leads to secondary metabolite production. Dual culture system. As I mentioned that the process can be divided into two phases. The first phase is biomass production and the second phase is secondary metabolite production. Now, each phase has its own specific requirements, and by following the stage-specific strategies, it is possible to produce large amount of biomass with an increase in the accumulation of secondary metabolites. And this was first used by Zeng et al. In 1977. Now, in these two phases, in the phase 1, the culture conditions, the culture medium, the culture conditions like pH, temperature, light, etc. would be very different from the culture conditions that are required in the phase 2, which is the secondary metabolite production. So now we would be discussing about the various factors which are affecting stage 1, which is biomass production. The first factor which affects the biomass production is the inoculum density. And we know what is inoculum. Inoculum is what we add to the medium so that the proliferation starts, right? So in this case, the inoculum would be the callus cells or a previously raised cell suspension culture. Now there is a critical minimum inoculum density below which cell or organ growth will not be optimal. So this stage needs to be optimized and it varies from species to species. We need to optimize the fresh weight requirement of the inoculum per liter of the culture. Coming on to the next parameter, which is culture condition. This would involve both chemical and physical culture conditions. This would include tissue culture medium and the physical conditions like light, temperature, humidity, pH. Now, talking about the culture medium, we know that in the biomass stage, when we want to increase the biomass, what we need is we need large number of cells. We need proliferating cells. In order to achieve this, we would supplement the medium with high concentration of inoxin, which is 2,4-D, so that the cells divide actively and reach a particular density before they can be transferred to the next phase, which is secondary metabolite production. So in this phase, there is high auxin requirement. This stimulates dedifferentiation and would at a later stage affect the secondary metabolite production. Then we also there are other physical factors which we also need to control, like what is the pH of the medium. There is an optimum pH at which the cells would divide actively. Then light also influences the culture conditions, and the temperature. The temperature range is typically between 17 to 25 degrees. And this is actually normally in tissue culture, as we know, the optimal temperature is 25 plus minus 2 degrees centigrade. So similarly, for secondary metabolite production, there is a range, like it is between 17 to 25 degrees centigrade. Now, coming to the next factor, which is selection of high yielding genotypes. Now, we know that during culture conditions, there are many changes which happen to the cells or to the organs. The reason being the culture medium itself kind of acts as an agent to bring about these changes. Now, these changes can be additions, deletions, and all kinds of modification. So as a result, our high producing cell line can convert to a very heterogeneous population wherein the secondary metabolite production will not be optimal. To achieve that, what do we need to do? We need to make the lines homogeneous continuously. This can be brought about by a technique called single cell cloning. So what are we going to do in this technique? In this technique, what we will do, we will take an inoculum from our suspension culture, plate it on our selection medium, which is solidified with a gelling agent, allow the cell colonies to grow, then each of the colony would be screened for the secondary metabolite production. The ones which are high producers will thereafter would be multiplied and taken into production. So this is a continuous process which one needs to do when one is undertaking trying to do large scale production of secondary metabolites. Now coming on to the fourth parameter, which is agitation. Now agitation is another important parameter which controls the cell culture, and this is irrespective of the scale of operation. It facilitates better growth by promoting cell conditioning, making the nutrients available to all cells, diffusion of gases into and out of the culture medium. It prevents buildup of carbon dioxide and other toxic exudates from cells, and the medium is sufficiently oxygenated. However, extreme agitation may have a very detrimental effect, because when we are agitating or shaking our culture at a very high speed, there is shearing of cultured cells. This shall also lead to reduction in the mean aggregate size, which is also very important, and the cell viability will be compromised. So to prevent this, plant cells are often grown in stirred tank bioreactors at a very low agitation speeds. Talking about the next parameter, which is aeration, and according to Georgiev et al. 2009, it has three main functions. First is maintenance of aerobic conditions, desorption of volatile products, because most of the secondary metabolites, they may be most of the cell cultures, they would be releasing some volatile products which may be detrimental. So when we are providing aeration, it leads to desorption of volatile products, and then it also helps in removal of metabolic heat. In certain cases, it has been observed that high oxygen concentration is toxic to the cell's metabolic activities, and it also may decrease the nutrient availability from the cultural broth. So aeration helps eliminate all these problems, and as we know, the carbon dioxide is an essential nutrient in plant cell cultures, so it also helps in equal distribution of all the required gases for the cell cultures. Coming to the second stage, which is secondary metabolite production. See, for secondary metabolite production, we need to change the culture medium. We have to make it more rate limiting, or in other words, growth inhibiting, such as we can reduce the phosphate levels of the medium, we can reduce the total nitrogen content of the medium, or we can add various adjuvants to the medium, which we would be discussing now. The first that we are going to discuss is about the precursor feeding. See, in a sense, what we are doing is we are kind of making the environment more challenging so that the cell growth is inhibited, and there is an element of stress for the cells which kind of forces them to start producing secondary metabolites. Coming on to the first parameter, the first strategy, which is precursor feeding. Now, we know that in a biosynthetic pathway, one biomolecule serves as a precursor for the next reaction. So depending on the metabolic pathway of the secondary metabolite produced by the cultured cells, precursors are added to the medium, which are then converted to the desired product by the pre-existing enzyme system in the cells. And in this, the timing and the concentration of the precursor is critical for production of secondary metabolites. For instance, Continental in 1999 reported, like they were working on Catharanthus roseus, and they reported an increase in secondary metabolite production by addition of certain precursors like tryptophan, tryptamine, loganin. The next strategy, which is that of elicitors. Now, what do we understand by elicitors? Elicitors are the biomolecules that stimulate the production of secondary metabolites, and the process is called elicitation. You can categorize these elicitors into biotic elicitors and abiotic elicitors. Biotic elicitors can be of fungal or of bacterial origin. Abiotic elicitors, they can be inorganic salts, heavy metals, some UV radiations, high salinity, high and low osmolarity, extreme temperatures, high pressures. So there are multiple ways by which you can, trigger the cells, put them into stress so that they are able to produce secondary metabolites. Advantageous as the cells release the product into the medium. And elicitors are mostly applied during the growth phase or exponential phase, when cells in which the cells are actively dividing. For instance, Smith et al. In 1987 reported an increase by 90% in production of a metabolite from Catharanthus roseus by addition of NaCl in the culture medium. Now, another strategy is biotransformation. Now, it is a process by which an exogenously added compound is transformed to a high-value product by the cultured cells. We know that plant cell cultures and enzymes have the potential to transform inexpensive and plentiful substances such as industrial products into rare and expensive products. So these are kind of producing something like metabolites which are very novel, which were being not produced in the cell lines normally. So we are adding some compounds which are being transformed into industrially useful high value products. For instance, in Catharanthus roseus, a very unique glycoside by the name of arbutin was produced when hydroquinone was added to the cell cultures. And this arbutin is extensively used in cosmetic creams as a lightening agent. Naturally, arbutin is produced in certain plants, but it was normally not produced in the cell cultures of Catharanthus roseus. Coming to the next strategy, that is immobilization of cells. We know that in vitro cell suspension cultures has certain bottlenecks for commercialization of the secondary metabolite based products. For example, they show unsynchronized growth of plant cells, non-viable product yield, genetic instability of selected lines, low shear resistance of cells, and intracellularization of the product. So this technique, immobilization, has the advantages, like it helps in extending the viability of cells in the stationary phase, and thus enables the maintenance of biomass over a prolonged time period. It simplifies the downstream processing if in the cases where the products are secreted in the medium, and if the high density within relatively small reactors results in reduced cost and risk of contamination. It reduces the shear stress, it increases the product accumulation, and it minimalizes the fluid viscosity, which causes mixing and aeration problem in cell suspensions. Now, it helps in increasing the cell density by at least 2-4 times, and it separates the cell from the medium. And so it is very much relevant in processes which are biphasic. As we have discussed earlier, that secondary metabolite production is primarily biphasic. it is divided into two distinct stages: biomass production and secondary metabolite production. So it helps in increasing the cell density. And the non-dividing immobilized cells, they are less prone to the genetic changes. Now, there are two methods by which the cells can be immobilized. First is the gel entrapment method, and second is the surface immobilization method. But the most widely used out of the two is the gel entrapment method, in which the cells, they are entrapped in specific gel or combination of gels, which are then allowed to polymerize. And the gels which are used for entrapment are alginates, agar, agarose, carrageenan. Likewise, the surface immobilization, it is another method that takes advantage of the propensity of culture cells, and the cells, they adhere to the inert surfaces which are immersed in liquid. And some of the examples of surface immobilized substances are polyurethane foam, fiberglass mats, unwoven short fiber polyester. So now coming on to the next strategy for secondary metabolites is permeabilization of cells. We know that plant secondary metabolites are largely produced intracellularly, and so extraction of such products into the medium is very important. And it is very important also because the removal of secondary metabolites continuously from the medium increases the productivity. It allows repeated use of culture, and in this permeabilization, using different agents, what we are doing is we are making the surface more porous or permeable so that the secondary metabolites are released into the culture medium. Now, this permeability can also be reversed. Now, some of the treatments that are used for permeabilization are DMSO. You can reduce the pH, make it more acidic. Sonication is another treatment you can use, or you can give electric shocks. Coming on to the next parameter, which is selective adsorption of metabolites. Now, it has been observed that the secondary metabolites which are produced in the culture, the cell cultures, they should be removed quickly. Otherwise, it would lead to processes like feedback inhibition, enzymatic or non-enzymatic degradation of the product in the medium, or volatility of the compounds produced. So it is very important to develop a separation technique that can concentrate the desired product. So some of the products which are used for the removal and sequestering of the product are polycarboxylic ester resins, neutral polymeric resins, and these can absorb these secondary metabolites. And in few cases, activated charcoals and RP8, which is a lipophilic carrier, zeolites, polyethylene glycol, cyclodextrins, all these are used to help in separation of the secondary metabolites which are produced so that the process is more efficient. Now, students, we are going to talk about another technology, which is hairy root culture. Now, why do we need to do hairy root culture? We know that secondary metabolite production in cell suspension culture has number of limitations, such as a cell population is not at all stable, they mutate, and thus the yield gets affected. And also we also know that the secondary metabolite production is highly localized in most of the cases. They are localized in certain organs. So hairy root culture is another technique by which we can override the limitations of secondary metabolite production in cell suspension culture. This process can also be automated, and we can achieve we can have high biomass of roots using transformation technique, in which we infect the tissue with a bacterium called Agrobacterium rhizogenes. We know that roots, when cultured under in vitro conditions, they exhibit extremely slow growth. So there has to be a mechanism by which we can increase the rate at which they are produced. So as I said, we can use this bacterium, Agrobacterium rhizogenes. How? We will talk about it shortly. The in vitro produced roots that we transform, they branch extensively and they're very genetically stable. and the growth rate is as high as 2500 to 5000, as has been observed in Hyoscyamus muticus by Flores et al. 1987, and it was observed that it is much faster than the suspension culture. Now students, we are going to discuss the method for raising hairy root cultures. First of all, we are going to take plant tissue. We'll take it from a genotype from where we have already analyzed that it has it produces high secondary metabolite. So we are going to take a tissue from that particular plant, we sterilize it, and then we induce some wounds and co-culture it with Agrobacterium rhizogenes. Agrobacterium rhizogenes is a bacterium which is used to induce adventitious roots from a tissue which does not have root initials. So it can induce roots from any kind of tissue. So after we have wounded and treated our tissue with Agrobacterium rhizogenes, we are going to incubate it under ideal culture conditions on the ideal culture medium, so that the tissue it starts producing adventitious roots. So once we see adventitious roots coming out of that particular tissue, our next stage would be excising that part which is showing adventitious roots emerging. We will culture it to a next medium which would kind of stimulate the tissue to produce more such kind of tissues or the roots. So once we have, and we are going to do it on a Petri plate, once we have good multiplication of roots, we are going to take it to then the next stage, which is the bioreactor. In the bioreactor, it is an automated system wherein all the parameters are controlled. The nutrients, the culture condition, the physical parameters, they're all controlled so that there is an optimal production of whatever we are intending to, whether it is biomass production or whether it is secondary metabolite production. So we are going to culture our hairy roots in the bioreactor and allow it to grow for some time, and once the biomass has reached to certain stage, we are then going to change the medium to a rate limiting medium so that it starts producing, it acts as a stress, and the root tissue starts producing the desired secondary metabolites. As you can see in this picture, it is actually in this picture you can see there is a production of hairy roots of ginseng. You can see the big reactors. First, the biomass is produced extensively. The roots are allowed to grow. The conditions are given such that there is high biomass production of the roots. Thereafter, it is then transferred to the second bioreactor, in which the conditions are made more rate limiting so that the root cultures start producing secondary metabolite. Thereafter, the entire roots are harvested. These are then used as such by little downstream processing. Students, in this slide, I want to highlight the benefit of carrying out secondary metabolite production under in vitro system. As you can see in this slide, there is a comparison between field cultivated ginseng and second between the roots which are growing in a bioreactor. Although the production cost is relatively high in case of the bioreactor cultivation, but if you see the yield, it is many folds. So in totality, the bioreactor cultivation proves to be much cost effective. Now, as I had briefly talked about bioreactors and you are aware of bioreactors, just to brush up your knowledge, bioreactors, they are the automated units which have all the quality controls for large-scale production of biomass as well as secondary metabolites exuded by the cultured cells or organs. and they have all the inbuilt system. There are all the controls are available. You can control the nutrients, you can control the aeration, the temperature, pH, light intensity, etc. And as we know, the culture is greatly affected by aggregation of cells, which eventually results in settling down of the aggregated cell, and this results in non-uniform availability of nutrients and oxygens, and eventually decline in secretion of secondary metabolites. So the bioreactors, there are many bioreactors which are there. Although more research is being done on improving the efficiency of bioreactors and for eliminating the drawbacks which the existing bioreactors have, but the effort is to control all the areas which actually is affecting the efficiency of the final product, which is the secondary metabolite production. Just to list the types of bioreactors which have been reportedly used, there are stainless steel stirred tank bioreactor, rotary drum bioreactor, bubble column bioreactor, trickle bed reactor, which is used for hairy root culture. Then there is gas phase bioreactors, spray droplet reactors and mist reactors, balloon type bubble reactors. So these are all automated units wherein there are all the controls available. Now each of the bioreactor has some advantage over the other. It has its own specific advantage which is there, and these are being used in their different case studies. They are being used for production of secondary metabolites. Now for commercial scale up, this is just to summarize what we have discussed earlier. Since the process is biphasic, a biomass production stage and secondary metabolite production stage, the bioreactors there can be two set of bioreactors. The first one, you can grow your cell suspension or hairy root cultures for large scale production, or you can say for biomass production. And the second bioreactor, the first bioreactor would be connected to the second bioreactor where the conditions would be made rate limiting. The stress level will be increased so that the cell or the tissues which are being cultured inside that reactor, they are forced to produce secondary metabolites. So in other words, you can say there would be the conditions would be more of growth limiting so that the production of secondary metabolites can happen. Some of the secondary metabolites which have commercial importance and which are commercialized or being produced at commercial scale are Taxol, Chiconin, Scopolamine, digoxin, berberin, morphine, quinine, vinblastine, atropine. So students, let us summarize what we have learned in this module. We know that secondary metabolites are produced under conditions of stress. In vitro production is a two-step process. First is the biomass production, second is secondary metabolite production. And each step has its own physical and chemical requirement. Although this process is very cost intensive, and it also requires a lot of research inputs across the production, but the efficiency is very high, and that makes the overall process very cost effective.